Dr. Chen and his team at the Houston Methodist Research Institute are helping to advance the field of organ transplantation with the identification of a troublesome subset of CD4 T cells, a subset that promises to be a more effective therapeutic target for preventing transplant rejection in patients. The molecular pathways leading to T cell activation have been extensively studied, but the mechanisms modulating subsequent CD4 T cell effector programs are not fully understood. Recent advances in the field include identification of antigen-primed stem-like CD4 T cells in models of multiple sclerosis and chronic infection. Studies have identified stem-like features of these CD4 T cells, including the capacity for self-renewal and persistence, and the ability to differentiate into effector cells.
Although T cell research has been extensively studied for 3 decades, we're still missing a very important picture to understanding T cell response. I'm Wenhao Chen, a PhD scientist in Houston Methodist Research Institute, and my work is mainly focused on T cell biology science in the field of organ transplantation.
To gain insight into the role of CD4 T cells in transplant rejection, Dr. Chen's study utilized single-cell RNA sequencing to analyze the CD4 T cell response in transplantation scenarios. Published in Nature Immunology, the results indicate the presence of stem-like CD4 T cells in transplant recipients, as well as the stem-like program directing the CD4 T cell response in models of transplantation. More specifically, they found stem-like CD4 T cells that recognize transplant antigens and can differentiate into effector cells that attack transplanted organs. Their data indicate that naive alloantigen-specific CD4 T cells develop first into effector precursor T cells and then into effector cells. However, the effector cells lose their ability to proliferate and fail to reject allografts upon adoptive transfer into new recipients. Conversely, the TCF1 high CD4 TEP cells can self-renew and differentiate into effector cells, thereby representing the stem-like CD4 T cells in transplantation scenarios. Crucially, the rejection of allografts is dependent on continuous replenishment of TCF1 negative CXCR6 positive effectors from TCF1 high CD4 positive TEP cells. The sustainability of the CD4 TEP cell population is largely dependent on TCF1, while differentiation into effector cells requires other factors, including the transcription factor IRF4 and the glycolytic enzyme LDHA. In fact, IRF4 or LDHA deletion in T cells resulted in transplant acceptance.
So that's our research goal for long term, is try to find a selective method to only target the T cells that cause trouble in organ transplantation, and leave the T cells against infection or other, for example, against cancer, leave them intact. In that case, we have to understand the T cell response in very details. Otherwise, we don't know which T cell is the good guy, which T cell is the bad guy. And that's our research focus during the last 2 decades.
The exciting results of this work have provided robust evidence for a stem-like program that controls the self-renewal and effector differentiation abilities of CD4 TEP cells. Because the stem-like CD4 T cells continually replenish the effector cell pool, they might be the more effective therapeutic target for preventing transplant rejection and in other T cell-related immunotherapies, allowing for the development of innovative therapies for patients with chronic infections, cancers, and autoimmune diseases.
In autoimmune disease, the naive cell become effector precursor cells. Effector precursor cells also extensively proliferate before they can differentiate to effector cells. So our work is quite similar to autoimmune disease conditions. That's I'm very happy that the field, the leaders in the transplantation immunologist field, they will recognize that. And certainly I believe that they will also contribute to this framework and try to target stem-like cell in their work as well. And I welcome to that. And the science is make progress. It's not one team to make progress. It's a whole research community pay attention to this framework and will make huge contributions.